Hi, welcome everyone to this next video. So in the last video we saw that we can model bacterial growth in a batch culture or in any environment using an exponential growth curve. But very soon we will find out, as we found out through the calculations done towards the end of the lecture, that very soon we will run into some problem. And the problem is that exponential growth facilitates growth so fast and the numbers increase so rapidly that whatever environment in which this growth is taking place will very soon prove to be insufficient to support growth of that large a population. As we saw through that toy calculation, that in a time as small as 3 days, bacterial population, if assumed to be exponential, increases to such an extent that its combined mass exceeds that of the mass of the planet. So clearly exponential phase, although very important, is only applicable for a short duration in the growth phase of a bacterium. So what we will do in this lecture is discuss two other models associated with growth, which allow us to some extent make more realistic assumptions about how growth is going to take place. So here in the slide is what we discussed in the previous video, exponential growth, and this was the problem that this shoots up to an unrealistic number. And that's a problem. What we are going to do today is a model called logistics growth. And what this model tries to do is it tries to learn from the physics of what is happening, the physiology of what is happening, and tries to incorporate that in a mathematical form. To understand what is going on, let us once again revisit the typical growth curve of bacteria in a batch culture. Let us assume that we start with n naught number of bacteria, and we saw that in this batch culture this is what is going to happen. And let us not worry about what happens beyond this, because really we are only interested in the period of time where growth is supported and the culture reaches stationary phase. Of course, many people study what happens in and after stationary phase, but let us stick our concentration to only lag, log and stationary phase, how a bacterial population arrives at the stationary phase. So this is what we know from experiments a typical growth curve is going to look like. Now let us try to imagine this in conjunction with what is happening in this flask. So here we have our flask, it is shaking, there are nutrients available, and we seed this with a bacteria. And this bacteria is going to divide, which is going to divide again, and so on and so forth. And this exponential phase is depicted by this exponential increase in number of bacteria in the flask. However, we note that there is a deviation from this curve as time moves forward. If this curve was truly exponential, then the number of bacteria would have moved along this trajectory. If it was truly exponential from n naught, this would have been the curve. However, what is happening here is that there is a slowdown in growth when we compare the actual growth of how numbers increase to a purely exponential growth. So this is purely exponential and this is actual growth. So if we compare these two, we note that exponential and actual growth dynamics only match each other for a small window of time from 0 to t naught, and beyond t naught the actual growth slows down and very soon it completely halts, and right here growth is, so there is no growth in this phase because numbers are stationary. So growth rate suddenly not suddenly, growth rate beyond a certain point deviates from exponential, slows down and keeps slowing down until it reaches just 0. So, let us record these observations and see how we can mathematically understand them. So, in the initial phase, what this tells us is that it is perfectly fine to assume that growth is exponential. However, this initial phase, let us say, goes from 0 to T naught. However, as we move beyond T naught, growth is no longer exponential, it is in fact slower than exponential. To an extent that after a certain point, it slows down to an extent that growth actually halts to 0. Growth completely halts and eventually reaches 0. And this slowdown, the extent of slowing down keeps increasing. So, the extent of slowing down keeps increasing as we move from time t naught to time t1, at which point growth is virtually undetectable in this growth media that I am testing this model in. And remember, all of this is for a batch culture. All right. So, these three characteristics is what we have to satisfy when we have to talk about a model which represents that exponential phase is not going to last for very long, but in fact actual growth will very soon be slower than exponential growth. So, how do we tackle this? Mathematically, incorporating these three aspects that we just discussed are done by only including one term in the model that we already discussed. So, this is dn by dt equals Rn. We already discussed this. This in fact leads to exponential phase, exponential growth. But we have an additional term here which is 1 minus n upon k. By just addition of this additional term in this model that we already studied, we can incorporate all three conditions that we discussed in the previous slide, that in the initial phase growth is roughly exponential, beyond a certain point it slows down and becomes slower than exponential, and eventually it slows down to an extent that growth reaches 0. All three of these characteristics are included in the mathematical form of bacterial growth if we just multiply our exponential model with this term 1 minus n by k. So, what does this term do and what does this term represent is what we will look at now. So, to understand that, let us first discuss what is this variable k. K is called the carrying capacity of the environment. If you remember, a few videos ago we did this example of thinking of E. coli growth in a flask whose volume was 100 milliliters. It contained growth media which is called LB, and we saw that after sufficiently long time of growth in LB, the number of bacteria in this flask reached to 10 power 11. That is because in a LB culture, 1 milliliter of the liquid media can support growth of up to 10 to the power 9 bacteria. So, 1 milliliter supports growth of 10 power 9. After 10 power 9 has been reached for every milliliter of culture, all the nutrients that are present in the flask are gone and there is no more nutrient left to support growth. So, this is the limit of the environment to the extent that it can support growth in this flask per milliliter. So, if you have 100 milliliters, then the total number of bacteria that can be reached is 10 power 11 and not beyond that, because the environment simply does not have sufficient nutrients to support more growth. This number is called the carrying capacity of an environment, that how many individuals can an environment support, and beyond it cannot go beyond that, because nutrients because simply it's run out of nutrients that it contains. So, how many individuals can an environment support is the carrying capacity of the environment. So, this automatically takes into account the quality of the media and the quantity of the media. For instance, if we have Lb 1 ml at 37 with shaking, then K is 10 to the power 9. However, if we have 100 mils at 37 with shaking, then the K associated with it is 10 to the power 11. If this was not Lb, if the carbon that I supplied was in the form of glycerol 100 ml with all these conditions remaining the same, then the carrying capacity of the environment will be 10 to the power 10, because it will not support growth to the extent that Lb can, and so on and so forth. So, K, carrying capacity of the environment, represents two aspects of it. Carrying capacity of an environment represents the quality of the media. For instance, in this case, Lb 100 ml of Lb supports more organisms as compared to 100 ml of glycerol. So, this is reflective of the quality of the environment that we are supplying. It also represents, it is also a function of the quantity of the resources present in the environment. For instance, if we have 100 mils of the same resource versus 1 mil of same resource, the K associated with them is also 100-fold. So, you multiply the resources by 100 and we get 100 times the carrying capacity of what it was when we only had 1 ml of Lb. So, carrying capacity is a function of quality and quantity. It is also a measure of quality. Not only means quality is not reflected by only chemical constituents of the environment. Chemical constituents. For example, it is not just the difference that here it is Lb and it is glycerol here, but also physical and physical environment of the physical conditions of the environment. For example, if I had 100 ml Lb but at 75 degrees Celsius, then carrying capacity of that environment would be 0, because E. coli would not be able to reproduce in those environmental conditions. So, it is important to take care of the fact that carrying capacity represents a carrying capacity of an environment as function of quality of that environment and quantity of that environment. So, that is the K here. This N and this N represent the same thing, which is the number of individuals at time t. So, this keeps changing with time as the number increases. So, let us now see that how does this equation capture the three conditions that we listed on the previous slide, which were again these three. Initial phase growth is exponential, beyond T naught growth is slower, and growth halts and eventually reaches 0 as numbers keep on increasing. So, we will spend some time on looking at how does this equation help us achieve that. Okay, let us write the equation again. dN by dt equals Rn 1 minus n by k. And let us assume that the environment that we are talking about is the following. It is LB 100 milliliters, growth is at 37 degrees Celsius, there is shaking, that is the condition. Initial conditions N naught, number of bacteria at T equal to 0 is just 1, and what this automatically implies that K is 10 to the power 11, because every ml of LB can support 10 to the power 9 bacteria. And let us, for the sake of simplicity, let us just say R is equal to 1. So, given these conditions, let us see how does this play out. So, when I start the growth experiment, I am starting with 1 bacteria, this grows, divides and 2, and so on and so forth, and then there is this what are called this branching process, and the numbers keep increasing exponentially until they reach carrying capacity. So, starting with 1, I reach K, and that is how the numbers in the population move as time increases, and this is number of bacteria in flask, and time moves in this direction. So, when we are first starting out, so let us divide this into 3 phases and try and see what happens. In the first phase, this is close to start of experiment. This equation would be dN by dt equals Rn times 1 minus n by k. However, we know that close to start of experiment means, this is important, that we are approximating this equation, but under these conditions close to start of experiment. We know that k is a constant for these given conditions, which is 10 power 11, and close to start of experiment n, we started off by 1, and then it will become 2, and then it will become 4, and so on and so forth. So, n is very small, let us say if it is the order of 10. So, even if n is 1, although we start with 1, so 10 is still 3 or 4 generations away, this equation becomes Rn 1 minus a very small number, which is of the order of 10, divided by 10 to the power 11. This is roughly equal to Rn 1 minus 10 to the power, this 1 gets cancelled, this is 10 to the power 10, and then we bring it to numerator, this is 10 to the power minus 10. This is roughly equal to Rn 1 minus 0.000000001, which is 0.999999 times 9 times Rn. And if you see that this is very, very close to just Rn, the difference between these two is so small, is much less than 1%. In fact, this is like 1,000th or 1/10,000th of a percentage, even smaller than that. So, the difference between these two is really small. Hence, close to start of the experiment, growth can be approximate. If we are using this particular formula, then growth can be simply approximated as Rn. This approximation is only valid if this is close to start of experiment and our starting numbers were really small. If our starting numbers were really large, then we would not be able to neglect this particular factor. So, at the start, so what this equation predicts is that at the start of experiment, if n naught is much smaller than k, then growth can be approximated as exponential. If we go back a couple of slides and see, this is what we had written here, that in the initial phases growth is exponential, is what I see from experiments that I do. So, this equation successfully captures that part of it, that growth in the early phase is close to exponential. Let us go to the next phase. Now, we will see when population is close to but still less than carrying capacity. Now, let us imagine a scenario where n is roughly k by 2. So, we saw that k was 10 to the power 11, that means n is approximately half into 10 power 11, which is just 5 into 10 power 10. In such a scenario, what you should note is that this fraction becomes roughly, if n is approximately k by 2, then this fraction is approximately half, because we get 1 minus n upon k as simply equal to 1 minus, instead of n I will replace k by 2, 1 minus k by 2 divided by k, which is just, the k's cancel, 1 minus half, which is just half. What that means is dn by dt is equal to r upon 2 n. So, this predicts that when the number of bacteria is half the carrying capacity, at that point population growth is still exponential, but the growth rate has reduced from r to r by 2, and so on and so forth. And we can see that if n was k by 3, then growth rate r effective would be not r but 2r by 3, and so on and so forth. So, as this n is increasing, the r effective is decreasing. In this case, r effective is just r by 2. This satisfies the second criteria that we were looking at, which is that beyond T naught growth is slower than exponential as defined by this particular exponent. Growth is getting slower as numbers are increasing. That defines the second criteria. And the third one says that as n approaches k, in this population, as n approaches k, growth should completely halt. And that's easy to see through this model, because dn by dt in that case would be, the equation that we have is dn by dt equals Rn minus 1, Rn times 1 minus n by k, but in the scenario where n approaches k, so n is very close to k, in that scenario if we look at dn by dt, that is just equal to r n 1 minus a number which is almost k divided by k. This fraction here is 1 minus effectively 1, hence it comes to 0. So, when n is equal to k, dn by dt is equal to r n times 0, which is 0, which means dn by, if dn by dt is equal to 0, this means that as t changes or as t moves forward, the rate of change of n is 0. As t moves forward, n does not change with time, because the rate of change of n is 0, which means that n remains constant with time. Once only this is only true once it reaches k. So, we have seen that by looking at this logistic equation, we can see that it satisfies what we actually see in growth curves happening when I perform experiments, that initially growth is almost exponential. As numbers increase, growth slows down and the exponent reduces, and finally as the batch culture that I am talking about reaches saturation, growth completely halts and approaches 0. So, just by incorporating this particular variation to the exponential equation that we started with, we can see that growth is now able to capture all these facets what I see experimentally happen. In this context, we can do one more thing, that imagine that I have species A and another species called species B, and I co-culture them together in the same flask. So, this is a flask which has been seeded by one individual of type A and one individual of type B, and our the challenge before us is that as time moves forward, how are we able to predict that what happens to number of A individuals and what happens to number of B individuals as time moves forward and this flask reaches saturation. A minor modification in the equation that we just studied will allow us to do that. So, in that case, we will write an equation that dNA by dt. So, we will write two equations here. The first one is for the first species, dNA by dt is simply equal to RA NA times 1 minus NA plus NB divided by K. Let me first write down both equations and then I will explain what these actually mean. Similarly, for the other species, dNB by dt is equal to RB NB 1 minus NA plus NB by K. So, what do these terms mean? The K here is the carrying capacity of the flask in which growth is happening. RA is the growth rate of species A. If species A was present by itself, this is how fast it would grow. Similarly, RB is the growth rate of species B. NA and NB are the number of individuals belonging to species A and species B, and in this term in the bracket here, we have to add both of them, because remember that K is the total number of organisms that the environment can carry. Those K individuals could be of species A, could be of species B, it does not really matter, but sum total of them cannot exceed K. So, the fractional capacity of the environment that has been used up is equal to Na plus Nb. This is the total number of individuals at any given point in time that the environment is already supporting. The total number of organisms that the environment can support is K. So, this fraction here represents what fraction of carrying capacity has already been used up. And obviously, when we subtract from 1 this particular fraction, it tells us that how much carrying capacity, how much fractional carrying capacity of the environment is still left in terms of resources being available to and support further growth. And if we solve these two equations together, and we will not get into that too much here, but if we solve these equations together, we can get the profiles of how Na changes with time and how Nb changes with time. And these have to be solved together because if you note here that this is an equation for Na, but it contains an Nb, and Nb is also a function of time, it is changing with time. Similarly, this is an equation for Nb, but it contains an Na, which is also changing with time. So, Nb is dependent on Na, but Na is dependent on Nb, and hence these have to be solved together to get the profiles of the growth of these two species together. So, in this video we have talked about one more approach of how to quantify growth and look at fitnesses of different strains when they are growing in a batch culture.